So welcome everyone to this month's version of the ReproNim webinar. We are super thrilled today to have a guest speaker, Neda Jahanshad, representing the ENIGMA Consortium and many other important efforts. Neda is a associate professor of neurology at the University of Southern California, and she single-handedly runs the ENIGMA Consortium. Oh, maybe not, but so she's an important influencer and lead influencer of the ENIGMA Consortium, and runs suicidal and aging brain projects of that, and will be speaking today on the evolving landscape of structured multi-site neuroimaging analysis. So without further ado, and I see Paul officially chimed in that my statement was perhaps accurate, that she single-handedly runs it. So it's I will turn it over to her to fill us in on the topics today.
Well, yeah, thanks so much for having me. It's always a pleasure to talk with you all and bounce around some ideas. I don't have anything to disclose. I will be talking a lot about ENIGMA, but I think before we get into some of the details of the projects and some of the findings, I want to go through what ENIGMA actually is and more what it's become. It's continuously growing. So it's become this dynamic consortium that's very collaborative, and it continues to organically expand both in scale and in scope. You might obviously, everyone would be very familiar with a lot of other large-scale multi-site data sets, ADNI, ABCD, UK Biobank, PPMI, and so many others. And these are tremendous resources. And you might ask, why would you want to pull any data or information from other international sites when there are these really important and very useful repositories available? Well, lots of imaging studies have been ongoing over the last couple decades. Some of them have completed data collection many years ago, but that's still data that's been collected, that's still information that's out there, and this information can still be used to help answer questions about the brain and its disorders, especially when it comes to studying rather specific, rare, or uncommon conditions around the world. And it really helps us answer questions about reliability of findings and generalizability across different populations. And we can actually, these aren't distinct necessarily. We can combine all these efforts together. And that's what we've been doing within ENIGMA to try and maximize the power for our analyses. So ENIGMA currently has, I didn't count the number of working groups, but there are quite a number of working groups related to both computational analyses, development of data analytics and informatics, and also neurobiologically organized groups that study specific traits or conditions. And these continue to grow as they're or divide sometimes. If we had initially one group on traumatic brain injury, we realized that it would be helpful to split these up into very more specific types of TBI. So the way these groups come together, I think this is something that's important to make people aware of. It's often left it's often led by one or two established investigators and one or two early career investigators who come together and realize that there is this network of common interest across a small group of their colleagues, and they'd like to try and expand that and work within the framework of ENIGMA. And oftentimes it starts with just a single project that expands to other projects. So, and what I mean by that is that the initial working group projects, like when a working group starts, really aims to grow the network, establish collaborations, and understand really the strengths and the limitations of each of the individual cohorts that get involved and participate. And because of that, some of the first projects ask very straightforward and broad questions that could actually be asked from all participating sites and cohorts. For example, in cases of certain conditions or diseases of interest, we might try and have the working group have as many contributors as possible that have data sets relevant to that disorder, and do a case control analyses across all the data sets. There is no requirement to sharing individual participant level data when joining ENIGMA, and that helps grow some of these initial projects and get groups and PIs comfortable with working in such a network. A lot of them might not be familiar with large-scale collaborations, and this helps sort of welcome them to the process and ensure their involvement. And when it comes to these analytic plans or projects that get proposed, these are discussed, agreed upon amongst all the contributors, and then they're documented, and then they're distributed once it's finalized. And they're often run with developed and tested analytical protocols and workflows that get distributed to the individual cohorts, because again, the data sharing is not a requirement, but the analytic sharing is. So the initial projects often help determine the scope and feasibility of future projects. As I mentioned, there is not a requirement for data sharing. So ENIGMA is not a data repository, and that's a common misconception. They have, there's a whole spectrum of how shareable different data sets from across participating sites can actually be. However, in this framework, any member of a working group can propose a project. This often starts after an initial project that sort of gains an understanding of the cohorts that are contributing. And these projects are approved by the working group chairs and then presented to the members, who can then opt in or opt out of participating in that project. And if the project does involve data sharing, then each cohort that would have to share their data would have to go through the cohort-specific access rules and requirements. And these projects are very collaborative. There are continuous updates and discussions on regular communications, whether email or teleconferences. And because we are trying to allow projects to be proposed, there are oftentimes very specific questions that working group members are interested in. So we really aim to try and structure the data and the processes and the subsets of data in various ways throughout ENIGMA. So we are working on expanding an ontology that can pull other existing ontologies together and work within this sort of ENIGMA framework. So oftentimes in these, in the working groups, we ask different cohorts whether they have collected certain variables from their participants, what the inclusion and exclusion criteria were for every site or every cohort. And we had built the, we had currently built the ontology more as a framework for how ENIGMA works and how projects are related and working groups are related and how cohorts can participate in multiple projects and how to capture that information and ensure that the cohort that, let's say within ADNI, for example, the subset of ADNI that participates in the free surfer analyses for Alzheimer's versus cognitively healthy might be different than the subset of ADNI that participates in the diffusion analyses that regress, let's say, ApoE4 genotype on FA measures. So these will be overlapping, but not exactly the same cohorts. So we've developed an ontology to try and distinguish these and ensure that we keep the hierarchy in terms of it's still all under ADNI, but involved in different projects and different subsets, and we can capture the project-specific information, like the mean age of the individual project, for example, or project subcohort. So that's what we've been focusing on, but we are able to pull in existing ontologies, and we look forward to working with groups here to do that. So pooling different diverse data sets is not easy. ENIGMA's approach has been largely revolving around the analytic harmonization. ENIGMA was launched to tackle the reproducibility crisis, specifically in neuroimaging genetics, and ensuring that we do have detailed and documented analytic plans are central to the workings of the consortium. And that involves the documentation of specific imaging metrics, clinical metrics, and quality assurance and statistical workflows. We know that the choice of analytic approaches can impact findings. That's been very well established, and there are numerous factors that can contribute to differences in the derived imaging metrics, even from the same scan. One thing that my group is actively working on is trying to see if we can quantify the degree to which those choices matter, and in what cases they might matter. If there are certain features for which, let's say, free surfer version would make a difference in the overall results. So, for example, Alyssa Azu and my group took a look at the diffusion imaging measures provided by UK Biobank. Now, I don't think there's anyone who actually wants to rerun 40,000 or however many they have now scans through another protocol, starting from the beginning, from pre-processing and extracting other measures. So how reliable are those measures if we were to compute the same measures using the ENIGMA diffusion analysis workflow, for example? How well do they correlate? And she found that for the most part, they're very highly correlated. There are a few regions that don't correlate well, but those were actually regions that we knew were not reliable from previous studies. Elizabeth Haddad in my group has done an extensive analysis of test, retest, and within and between version compatibility, reliability, and compatibility across free surfer versions, which we use a lot in ENIGMA, as there are some plans now to move to the latest version. Initially, when ENIGMA started, the latest version was version 5.3, so most of the cohorts had run their images through 5.3, but now would it make a difference if they ran 7.1? Would that be necessary? We're finding there is quite a bit of compatibility, but there are certain regions and certain metrics, especially in the cingulate, that are not compatible at all. And I just wanted to thank you all at ReproNim for for working on ways to capture these workflows and allowing us to have this data provenance that we need. And we're really looking forward to working with you on capturing some of these workflows within ENIGMA. You might be, this might make everyone a little bit nervous, but our ENIGMA's very first project was a GWAS of hippocampal volume, and ENIGMA had not taken off. We did not know whether we could convince people with large data sets to reanalyze their images. So you might notice that it says in small print in the methods section at the very end of the paper that each site was permitted to use any valid automatic segmentation algorithm. Now that's something that we're moving away from now, and we're trying to maintain consistency across these workflows. But I think this actually is also important in itself, as the metric we were trying to estimate the hippocampal volume overall, regardless of whether FSL was used, free surfer, we don't know which version was used, or other methods. There was even a group that had manually segmented their hippocampal volumes. Regardless of this heterogeneity in the methods, we did have some exciting results that I'll go into in a second. But just to sort of ease your mind a little bit, we did do extensive test-retest between free surfer and FSL. We did find that they were largely well correlated, and we had distributed some harmonized protocols that could be used for quality assurance. So we did try and ensure harmonization on the quality aspect. Now, despite this heterogeneity and this variability, ENIGMA's first paper, we're calling that ENIGMA 1 in this slide, the most significant SNP, the top SNP that was determined to be to reach the threshold for significance after replication, ended up continuing to be the top SNP in further iterations of ENIGMA work. In a second International Genome-Wide Association study, we ran a GWAS on not only the hippocampus, but other subcortical structures, or the volume of other subcortical structures as well. And again, with the hippocampus, even though we had nearly doubled our sample size, that same top finding was significant and became more significant. And then again, when we merged those results with the results of the CHARGE consortium, we saw that it became even more significant. So despite that heterogeneity and the methods, we found something that continued to be robustly identified in different iterations. And to further prove that it did work, we saw actually that if you plug in that top SNP into the results of the UK Biobank GWAS, you'll see that across all the, I think maybe about 4,000 features that they run associations with, the top, actually the only significant phenotypes were the left and right hippocampus with either fast or free surfer. And if you then look at the GWAS, at the full Manhattan plot GWAS results of these, you notice that the top hit is again the same one that was found with not only diverse methods, but all of these distinct cohorts around the world. So that was pretty encouraging. And I think that now that we know that these meaningful and reliable results can be derived from pooling information across these diverse data sets, we can expand upon the analytic plans that we have. So after that initial 2012 publication, we developed and documented further ENIGMA protocols for additional imaging features. And the consortium quickly grew beyond genetics to include different disorder working groups, such as schizophrenia, major depressive disorder, all of which could use the same analytic protocols. And now more and more working groups that are studying very specific conditions are able to use the same protocols, and we can essentially look at results across these disorders in a systematic way. And not only are we looking within ENIGMA across these disorders, but actually there have been other consortia that have used our protocols, and we can now look across consortia to compare findings. So in, I think, 2018, we published case control effects of schizophrenia on the white matter microstructure using our ENIGMA DTI protocols. And this was later also done within the Cohort consortia in Japan, which is a multi-site Japanese consortium of schizophrenia patients and controls, and they also collect diffusion imaging, and we could see the results lined up almost perfectly. So we are expanding a lot of the imaging methods that we have. We are aiming to harmonize this feature extraction with detailed documentation, containerization. I know we've worked with David, you and your team, to containerize the so-called morphometry protocols. There are new efforts in the resting state processing pipelines with Leah Walker and Waller and Ilya Veer, called Half Pipe, that makes use of a lot of existing workflows, including fMRI prep, but expands beyond that. And now there are a lot of projects where the pipeline does ask the members to reanalyze all the data, and not just say we'll work with whatever you have, but we really are trying to reanalyze the data in a consistent manner. And oftentimes that does include the containerized workflows, in order to minimize the vast methodological variability that we already have from the scanners themselves. And that allows us to ensure reproducibility on so many levels. One of the core protocols that we redistribute is related to the quality of the features that we're trying to extract. We know there is so much variability in the data that gets used for different ENIGMA projects. We want to make sure there is some level of visual quality assurance across any feature we're trying to extract, whether and especially across different stages of the processing pipeline. So, for example, there are subtle errors in FreeSurfer that we will make an extracted feature not truly reflective of the anatomy it's supposed to be reflective of. So we've documented a quality control guideline that will allow users to go through and see if they do have errors in their segmentations or in the cortical parcellations. The half-pipe fMRI protocol has a nice GUI with it that allows you to go through each one and rate them as with smiley faces, whether very happy, sort of mediocre, or bad. And a newly distributed workflow for cerebellar lobulars also contains these quality control assurance metrics. So this is something that we generally include within our distributed protocols, is this one aspect of manual intervention and just checking, really. At the same time, though, we are trying to develop methods that allow this to go a lot faster. So you don't have to, try to go through tens of thousands of screenshots of these outputs. We've already had to go through tens of thousands of these manually. So now we're using some of the metrics that we've been able to extract and our pass and fail guidelines to try and automate this and provide at least some filtering of at least images that you probably don't need to check for quality. And Shruti and my team has been developing this, and if you'd like to test it out, please do so. It works well for FreeSurfer 5.3, and we're working to improve it for 7.1 now too. So now features have been extracted and quality controlled consistently. Now we want to actually use those features for a particular project, for a particular study and finding. And as you see through the ENIGMA name, it all really started with meta-analysis, and that would be all groups running the same statistical associations locally on their end and sharing the summary statistics for analyses. So that was what was pulled as the summary statistics for the meta-analysis. This sort of is a workflow describing the process. So about 138 GWASs were conducted at all the 60 participating sites, and they had some level of quality control, but they sent those files, the quality control files and the results to the central location for the project leads, and those were quality controlled again. and we were able to visualize some of the results for a particular SNP, for example, with these forest plots that allow you to see the effects across each of the cohorts. And a couple more things and get a science paper. So that is just sort of the workflow for the meta-analyses. I won't go into the details of the study, but as with the GWAS, the same procedure was conducted for these various disorder working groups for these case control analyses, looking across cortical thickness metrics, and for these diffusion analyses as well that I mentioned earlier within, for example, the schizophrenia group here. Now we developed a way to also make these statistical scripts and analyses plans not only available to someone within the working group that's running that analysis, but available to everyone. So we have this co-mapper system on ENIGMA's GitHub. So this is for ensuring that you can easily run the statistics. You could easily look into exactly what variables are run, exactly how many linear models were conducted. Sometimes we would want to run the analyses in the full group and do follow-up analyses in, let's say, males only, females only. Like, these were all part of the analysis plan that was approved. And when the scripts were written and distributed, it's a very intuitive system for looking to see exactly what the variables were, exactly how the models were run, and how many models. And those could be used by another group to follow the same procedure, and same scripts could be used again with just a different input for knowing which analyses you want to run. So, but most of that was for meta-analysis. But now that these features have been extracted and members of the working groups are enjoying working with each other more, and they realize that they might be able to share these derived feature sets for some of the cohorts. Then not all of the cohorts can share the derived imaging features, but many of them were able to, and we several members of ENIGMA worked to compare findings from meta-analysis with those of mega-analysis, including all the features at a centralized location rather than just the summary stats. And this allowed us to look more in depth, not only within a study, but also across studies. For example, the study that was able to not only look at the effects of a particular disorder, like OCD on subcortical structures, but look across these three working groups at the effects and break this up into age bands, for example. So looking at pediatric, adolescent, and adult effects across these disorders. And that was really only possible because they were able to merge the derived subject level information together. And of course, that leads to statistical harmonization of these derived metrics, which we in this case that I'm showing here within the ENIGMA Epilepsy group in their GTI analyses, there were several subtypes of epilepsy that we wanted to look at the brain-wide effects and see what the effect sizes were for each of these subtypes across the white matter. But some of them are more rare than others, and really the only way to do a well-powered analysis of such a study was to combine the data. But the diffusion parameters are so varied across these different working groups that really we had to do the statistical harmonization, in this case combat, to be able to get meaningful effects. And you can see in this work before site correction with combat, really there are numerous outliers, and the distributions of all the different sites in these different colors did not overlap well, but obviously after harmonization is a lot more consistent. And this pooling of the metrics allows us to look at these more specific analyses. So in the Parkinson's working group, we recently published this paper that where we not only looked at the effects across all the Parkinson's patients compared to the matched controls, but we were also able to pool together scans of patients at different stages of disease according to the Hone and Yar staging, and see the progression of effects with stage. And this actually revealed something very interesting, at least we think so, where early in the disease stage, we, in this neurodegenerative disorder of Parkinson's, we see this higher thalamic volume early in disease, where we see later in later stages of the disorder, the volume is less than that of controls. Now, one interesting working group is the ENIGMA Neuro-HIV working group, where we're looking at the effects of immunosuppression, viral load, and these immune markers of individuals that have HIV infection, and assessing their associations with brain volume. Now we noticed a shift sort of in the profile of people living with HIV, where those that are not on medication, not on CART, have a more of the classic basal ganglia signature of HIV, of neuro-HIV, whereas those who are being treated and are more living with HIV as a chronic condition, we're seeing the effects of viral load and immune markers such as CD4 in the hippocampus and in other limbic structures. So one reason I'm bringing up this study is because harmonization within ENIGMA HIV is actually a little bit tricky, because there is such a strong confounding effect of site or cohort and different demographic variables. For example, we assessed these associations in both male and females, and found most of the effects were only in the males. But at the same time, if you look back into our demographic spreadsheet, nearly all of the females, not 100%, but something like 80%, were from a single site in South Africa. And those individuals also tended to be the ones that were largely not on antivirals. So there's these confounding effects with site and demographics sometimes, and harmonization on a statistical level might do quite a bit of overcorrection. So Mengting Liu in my group has been working on image harmonization with a style-encoding GAN, a general adversarial network, or where we are trying to match the style of a certain scanner's MRI image and apply that to any other image from any other cohort. So we have this model that's been trained, and we'll hopefully see it on GitHub in the next couple of weeks, but we are seeing it does a really nice job with image harmonization and even features extracted after the harmonization, and does reduces bias compared to combat. And the last quick thing I want to briefly mention is that a lot of these ENIGMA studies have been completed, but there are a lot more data that's also being collected that could contribute to studies that have been published already. So we're working on the system of continuous analysis, where we have the questions that we want to ask, and we can continuously search for data that will allow us to that work with that hypothesis, that will be testable. And we have this sort of continuous system working with Professor Yolanda Gill at USC. I'm not going to get into the details. We're calling it NeuroDisc because it is something that we can use within ENIGMA and also in other cases outside consortia as well. It has multiple parts, and it links back to the ontology that I described earlier, where we are trying to describe the individual cohorts and the level of information that they have, and specifics about the subsets that might have been involved in certain projects. So just as proof of principle, we were able to use the data that was provided in the supplementary tables of our Cortical GWAS work published in Science last year, and replicate the forest plot almost identically. What we did, instead of sorting the cohorts alphabetically, you'll see the order is a little bit different. We sorted here by age. And this system also allows us to go above and beyond the original analyses. We've implemented now a meta regression analysis, where we're one of the properties of each cohort is their mean age, and we're able to regress the effect size on mean age as well. So that's just one example, but we could take any other properties of the cohorts and do the association. So that is really how ENIGMA is expanding, and we are continuing to expand as new working groups and new projects are proposed. There are new ideas that require new algorithms and a new framework for all this. So I'd just like to thank about 2,000 people that I won't name that are involved in any and all ENIGMA projects and really help motivate this work and come up with these ideas. And thank you. Any questions?
Thank you so much. That was a tour de force of so many things that your initiatives have really supported. And again, it really is the state of the art in these big, large scale data sharings. Despite these other initiatives, again, I think this preceded them and really got people moving in the right direction. So thank you and Paul and the leadership team here for pushing that forward. We'll see what other people have for questions, but the thing struck me just early on in your presentation was your being anxious about saying, oh, we let people do their own thing. But actually, to some extent, especially now that we know that the different tools and different versions of things actually do affect things to some extent, I think the results that survive different tools, and if it's a volume of a thing or a thickness of a thing, and if that's really the thing that's happening, it really shouldn't be super tool dependent. And things that are super tool dependent make me more nervous about the tools than they do about the biology that they're describing. So there's some level of, I think you were ahead of the game and also lowered the barrier to participation by taking that approach, but also we're able to discover things that were large enough, the big enough effects, standard enough effects that it didn't really matter if you drew it by hand or if you used fancy software. So again, I think hearing you present it this way, that just struck me as actually being quite important to let that variety of tools and procedures.
I completely agree, and I think that's something that I know Theo has brought up as well. Is we want to make sure that any finding is not specific to the software that we use, and that is one of the reservations I often have about forcing a very specific pipeline on people, but I think ensuring people know the different components and that maybe it might not work for their data, and having that quality assurance and making sure that when we're trying to look at the hippocampus, that software is actually extracting the hippocampus in their data, I think is a step that helps ensure it's not too random whatever software is being used.
Yeah, thanks, Neda, and thanks, David. And sorry, I look like I'm in a darkened room. I'm sitting in a hotel in Chicago, so that's why it looks a little odd. Again, thanks, Neda, for a great presentation, and it's a great tribute to all the work that you and your colleagues have done. I wanted to ask you, and maybe also David and the ReproNim team, about the question of an updatable publication or a living publication. I know, David, you've emphasized the benefits of this and the need for it many times, and I think that some of the tools that you demonstrated show examples where new cohorts are added. And I don't know that you necessarily change your mind, but the balance of results is different. And I just wanted, Neda, if you could comment. I mean, are there some examples where, as a new flood of data comes in, there are informatics tools to sort of redo either redo parts of the analysis that are necessary, or sort of in a limited way redo the analysis. And maybe, David, this is also a question for you and your team. I mean, there are also not just informatics, but sociological issues with journals, because obviously the winning publication is the one with the highest N and the greatest rigor, but then if there's another one after that, there sort of needs to be a way for the field to see that the data have changed. I mean, just to give a silly example, I mean, the evidence that the Omicron variant of COVID is very bad might be different next week than today, and having a sort of nice curated literature and way of presenting evidence evolving. I mean, that's a ridiculously severe case, but even in neurology, I mean, the evidence for different things, particularly candidate genes, has changed, and not all the readers of the paper are aware when the evidence has changed. So I wonder if, Neda, I mean, are there sort of nice examples where we could sort of update prior publications and sort of demonstrate to the field that that's a sort of nice evolution of publishing? And I know, David, you've sort of championed this concept also. I think it's a very sort of liberating one for publishing.
Yeah, I've championed it, and I see that Neda and Yolanda have done it, so that makes me happy.
We're working on it. Yeah, I think we're using the the GWAS results of the cortical analysis for now as our sort of piloting example. So we're now adding, so at the time that was published, we had only accessed about 10,000 of the scans from UK Biobank. So now what the plan is for this paper that we're almost done writing up is to remove that cohort from the meta-analysis and replace it with the larger UK Biobank cohort now available, and just show how that updates the results for a single SNP. So we actually don't have GWAS level results for all the cohorts for this work. We're gonna request that after publishing this and sort of demonstrating the utility of it.
The other thing that, again, it's easy. I promote this every time I get the chance. And again, thank you for reminding me and you know, do the work you've done on that. But again, I think it's important both for, you know, any finding that we have, you know, there's always these chances of, you know, underpowered false positives appearing. And unless we have a way to, you know, follow them in a little bit more consistent way, it's just too easy to wish away, you know, or ignore, you know, things as more data comes available. So the false positives don't get damped down. But equally important, you know, our technologies, you know, evolve. And again, that's something I've looked at recently. Some of the things we didn't see in the initial, you know, NIH pediatric cohort of, you know, 300 or so done back in the early, you know, 2000s. Well, just because they didn't find it there, you know, the imaging evolves, the analysis evolves. And so to keep, you know, tracking down even the negatives, you know, because some of those don't stay negative because our technology and our cohorts, you know, evolve. And again, it's just so critical to have tools that, you know, let, you know, an investigator find, you know, put the flag in the sand. This is what I saw when I did it this way in this cohort, but that's not a permanent thing. And so we need to embrace, you know, that dynamicness through these types of things. So, JB, I see you have a hand.
Yeah, and it was such a amazing talk. So much stuff there. The one thing that among many other questions I would have is, you know, you seem to be really uniquely placed to get some of the harmonization across many variables, you know, and across many, you know, sites and things like that. And I think we're still a bit stuck in that respect. I mean, you know, many people are working on it, and like it's still like a bit of a jungle, you know. And I was wondering, what are your thoughts on how best we could sort of like, you know, hurdle the cats and basically, you know, organize our fields and say, okay, we're going to adopt those labels and those variables and those and maybe like this dictionary, this, you know, whatever is the right tool. So, I mean, we are proposing ours, of course, but like, you know, there's so many, you know, proposals and like, you know, and what do you think would be the best way to go forward?
It's a complicated question. It's such a mess. Like as you know, like each data set was collected for maybe a particular study, and they never thought of combining it with something else. There are so many steps involved in trying to merge. Like you said, from what I've seen, it seems to work when we focus on just a particular aspect that we're trying to, at that very moment, analyze. Trying to do it all, I think, is too much. And then you know, might want, when you actually go to use that data, you might want to do something else with it, and those labels won't exactly line up with what you need. But one example I'll give is in our suicide working group. What it is a combination of cohorts that study a variety of psychiatric disorders, and each cohort might have used a sort of disorder-specific set of assessments, but we're interested in suicidality, which is common across psychiatric disorders. So one of a few of the members within the working group pooled the different sites to see how many of them might have multiple metrics or multiple types of assessments on the patients and the samples that they collected data on. And they did an extensive correlation between each pair of examinations to see which ones actually are correlated, which ones could be represented by the same construct, which ones aren't actually, and trying to dig into that further to know whether it's more disorder specific or just a noisy metric is hard to know. But right now we are planning on using these assessments of suicidality for the next project. So it sort of has that sort of utility where it's being used now rather than 5 years from now where we don't know if it will be useful. So I think that answers your question. Starting focused and small might with the experts.
It does a little bit. I mean, there's so many ways of trying to solve that problem, but that's such a huge problem that I think it's a kind of community problem. Thanks, Neda. That was such a nice talk.
Thank you.
I know we're coming up on the edge of our official hour and some people are having to drop off, which is fine. I want to get on the record that next month we will be meeting on January 7th and Dorit Kleeman from University of Iowa will be with us talking on some topic related to autism, I believe. So stay tuned to your channels to get updated on the details of that presentation as that gets closer. Thank you again, Neda, for joining us and this lovely presentation. And thank you and your team for being a collaborative project and participants with the ReproNim initiative. And it's always great to work together.
Yeah, thank you. Take care.